SARS-CoV-2 and COVID19. I shall be discussing some aspects of the pathology as related to the disease of COVID-19. This will include some discussion of some journals, papers from journals, some research from the internet, and discussion of histological slides from autopsy material. Subtitle of this talk is Eye of the Storm. I am not a medically qualified doctor. No advice is being given. All information is for educational purposes only. Please read the disclaimer before continuing. Pause the video if need be, and then you may like to continue. After reading the disclaimer, you may like to continue with the rest of the video. My name is Harsh Sharma. This is a histological slide of a lung, a healthy lung. Very little tissue, well organized it is, and lots of empty spaces which would be filled with air in normal times. Each one of these structures is alveolus, 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 and together they make plural is alveoli. So each singular is alveolus and plural is alveoli. Each alveolus has a alveolar wall made of very thin epithelial layer, and the epithelium is made of type 1 pneumocytes, which are very thin, flattened, and occupy most of the alveolar wall epithelium. So alveolar type 1 pneumocytes are occupying most of the surface area of the epithelium. Then there are the type 2 pneumocytes, which synthesize surfactant, although they do not occupy much of the surface area of the alveolar wall. They are in fact more numerous. There are more type 2 cells than type 1 cells. So there is a wall of the epithelial layer of this alveolus, then there's one of this alveolus. In between the two walls, there are capillaries and basement membrane. There are other cells, but at the moment I just want to talk about the two main players, which is type 1 pneumocytes and type 2 pneumocytes. This is a histological slide from an autopsy material. That nice epithelial layer in the normal lung, consisting of type 1 and type 2 pneumocytes, is no longer there. This is an alveolus, not very empty alveolus in good health. This alveolus is not very healthy, and in this stage the alveolar epithelium is just not there. These, I would guess, are remnants of type 1 pneumocytes which have desquamated, they have come off the wall. I am not a histopathologist. All this is a new learning experience for me, but I can guess that these are in fact type 1 pneumocytes which have come off. A lot of capillary congestion. There is. This is just one aspect of histophysiology that occurs in these patients. Type 1 pneumocytes are no longer there. They are not, at least, where they should be. Some around here fallen off, but where they should be, there aren't any. This is a type 1 pneumocyte immunostaining, specifically for type 1 pneumocyte, which has desquamated. What about type 2 pneumocytes? Not only that type 2 pneumocytes have survived the ordeal, they are in fact proliferating. So there's a pair of proliferating type 2 pneumocytes. Here is one that is most likely type 2 pneumocytes which have gone on to make a multinucleate giant cell. So when they start proliferating in such circumstances, type 2 pneumocytes can form multinucleate giant cell. This is what it most probably is. Type 2 pneumocytes can differentiate to become type 1 pneumocytes. Logical hope is type 2 will differentiate to type 1 pneumocytes and repopulate the alveolar wall. So there they are, type 2 hopefully will differentiate to type 1, and there will be enough type 2 left as well to repopulate this alveolus, to give it a new alveolar wall. That is a hope, I suppose. But SARS-CoV-2 replicates in type 2 pneumocytes. So, will proliferation of type 2 pneumocytes lead to healing or provide more cells for the SARS-CoV to replicate in? This is immunofluorescence staining. The white is not just default white color, it is in fact fluorescent white light. Wherever there is this napsin, the experiment has been set up with immunostaining that there will be white fluorescence. Napsin is a molecule found in type 2 pneumocytes, so white fluorescence designates this cell as a type 2 pneumocyte. The red is SARS-CoV-2 RNA. So again, the experiment is set up that SARS-CoV-2 RNA will fluoresce red. And here is a cluster of type 2 pneumocytes with a virus replicating inside them. And they are pneumocytes because lots of white staining, white immunofluorescence rather. So again, a cluster of pneumocytes in which SARS-CoV-2 is replicating. But there's green. What is green? Lots of it. There's lots of green. Plenty green. What is it? The blue is, I think, nuclei. I'm going to call blue that they're nuclei. I'm a learner in histology, histopathology. Very early stages of my learning. So yes, I think blue are nuclei, definitely. But the green, what is green? Lots of it there. Interleukin 6, that's what green is. These type 2 pneumocytes are synthesizing a lot of interleukin 6. So our story begins with type 2 pneumocytes infected with SARS-CoV-2, producing IL-6 and proliferating. Please note there is a change of color in this scheme. In this slide, the virus RNA is labeled with green immunofluorescence. There are the airways. Swarm of SARS-CoV-2 virus released from lyzed cells. Just a swarm. So much of it there. The bright dots are in fact virus infected type 2 pneumocytes that have yet not been lysed. This is another aspect of pathology of many patients who became severely ill with SARS-CoV-2, COVID19, the disease. They are thickened deposition, waxy, glassy, shiny, and they are called hyaline membranes. So these are hyaline membranes. One in this alveolus, and effectively an independent deposition in this alveolus here. Plenty of it. Hyaline membranes. How do they form? Well, during the disease process, these alveoli actually became filled with fluid, protein-rich fluid with protein in it, cells, cell debris, surfactant, immune cells, macrophages, whatever there was in the lung, plus the debris in that liquid. It dried up and became hyaline membranes. Does this hyaline membrane contain virus particles and remnants of it? We saw so much virus there was. So, when these membranes formed, do they contain virus particles? This is an electron micrograph of hyaline, and these are what the authors say are in fact virus particles. I have seen a lot of electron micrographs of SARS-CoV-2 virus particles, but I have not yet seen any that confirms that these are actually virus particles. They most probably are, but scientifically speaking, there should be confirmation with some technique as immunogold electron microscopy. I have not yet come across SARS-CoV-2 virus particles, electron microscopy that is, where they have actually been identified as such. Morphologically, appearance-wise, size-wise, they seem to be virus particles. They seem to, in other pictures I've seen, seem to have spikes and everything, but nothing such as a specific technique that identifies them positively as SARS-CoV-2 virus particles. We need immunogold electron microscopy, please. These are two different samples from two different patients. Both are not to scale, but there are important issues to be discussed here. So there is the hyaline membrane of this alveolus. There is the hyaline membrane of this alveolus. Look, they're still close together. The stuff in between them is not very thick. Maybe a bit more thicker than normal. It would be in normal times. But in between, it's nothing like what is here. So, it is reasonable to assume that this patient was at one stage like this and then went on to become this state. What is happening here is immune cells have infiltrated in here, into the septum. And this infiltration of immune cells plus other processes going on. There is fibrin deposition. These dark pink areas are fibrin. They're more darker. They're more pink than this pink background. Okay, so these darker pink areas are fibrin deposits. Immune cell infiltration has thickened this septa. So it is reasonable to assume that this was once at this stage, and with time it has developed into thickened septum. Again, fibrosis, infiltration of immune cells that have infiltrated here to thicken the septa. There is the hyaline deposition. So this must be the alveoli. And thickening of the septa has in fact made the alveoli smaller, because the septa is now effectively bulging into the alveolar space. So it's reasonable to assume that within a patient or in an area of a lung, this kind of this development has taken. It's reasonable to point out this kind of thing probably doesn't happen in all of the lung. It only happens in some areas, allowing many patients to recover. Okay. These are questions, not statements. Is formation of hyaline membranes evidence that edema has gone and insult causing the edema has disappeared? If not, why did edema go? If yes, are the virus remnants within hyaline membranes inciting immune attack or inflammation? So, what question could be, why did the edema go? Is it because whatever caused that edema, whatever was causing it, has now been dealt with by the body, and edema, then the liquid had dried up, normal liquid was coming in? Or something else is happening. Maybe there's a new disease process taking place. Maybe these immune cells have come in to attack virus particles within the hyaline membrane. Many things could be happening, not just one. This is just one possibility. In this slide, brown is the surfactant and red is the SARS-CoV-2 protein. So here is a virally infected pneumocyte with red SARS-CoV-2 protein. This brown, what is this? What could this be? My guess is that this is surfactant embedded into hyaline membrane. So the liquid here contains surfactant. When it dried up, it became hyaline membrane, and it has been labeled with chemicals that label the surfactant as brown. In this slide, virus protein is stained red and macrophages are stained brown. So here's a macrophage stained red with virus protein inside it. There is a pneumocyte stained red, virus protein. Brown macrophage with red virus protein. Pneumocyte type 2 with red virus protein. Here's another one. That's a pneumocyte infected with a virus. Look at the morphology of it. This is virus cytopathic effect. So when the virus infects these cells, it causes a change in morphology to take, which is known as virus cytopathic effect. Early on became another effect of hyperproliferation, that was multi-giant nucleate cells. So here are macrophages in a type 2 pneumocyte. This and juxtaposition of macrophages with type 2 pneumocyte is not a chance encounter. I'm 99.9% certain these macrophages have actually come looking for this infected pneumocyte. They found it and they are now dealing with it. 3 macrophages around one infected pneumocyte. It is some kind of a process going on to bring them here and deal with it. So there's a possibility that macrophages are producing cytokines onto the pneumocytes, as well as trying to kill it. They're causing the pneumocyte to release further cytokines, which may or may not affect the behavior of the macrophages. All kinds of possibilities are there which need to be investigated. They may be producing, this pneumocyte may go on to produce cytokines, which may affect the behavior of other cells. Local concentration of cytokines may be much higher than peripheral. What is local? The whole lung, lobe of a lung, airway, several alveoli, just the air sac, just one alveolus, just the wall of the alveolus, just the junction with the capillary and the alveolus, a group of cells. Is that local? We need to study the immune response in context of different localities. Summary slide. Many of these samples came from this paper. IL-6. IL-6 has many actions, can do a lot of things. IL-6 is produced by many cell types, cells of the immune system as well as other cells. IL-6 acts on many cell types, cells of the immune system as well as other cells. Lots of things. Elevated in the adipose tissue. Megakaryocytes produce IL-6 and have receptors for IL-6. Plays a role in hematopoiesis. Potent effect on thrombopoiesis. Here there are lots of cells that produce IL-6. I would guess there's more. And then IL-6 acts on lots of cells. Again, I would guess probably more. Look at this. B cells produce IL-6. IL-6 acts on B cells. Endothelial cells produce IL-6. IL-6 acts on endothelial cells. We'll be talking a bit more about this. Just a reminder about autocrine, paracrine, and endocrine. Autocrine is when the cell produces the interleukin in this case, and then interleukin acts on the cell itself. Paracrine, cell produces the interleukin and it acts on a cell nearby. Endocrine, cell produces the interleukin, it acts on a cell far away. Well, not that far away, still within the body, but relatively speaking, much more further than this one. Receptors on cells for IL-6. There are two types of systems, receptor systems on cells. All cells have this type, this arrangement. Only few, namely some leukocytes, hepatocytes, have this arrangement. Here there is a membrane receptor for IL-6. IL-6 comes and binds to this receptor. Free IL-6, must point out. IL-6 itself on its own, alone, not holding hands with anybody, just by itself, comes and binds this membrane receptor. This then joins up with membrane bound GP130, then there is signal transduction. All cells, including the leukocytes and hepatocytes, so all cells have this arrangement where there is no membrane bound IL-6 receptor. What there is a soluble receptor in the blood, in the serum, in the body fluids, which binds IL-6. The two bound together, holding their hands together, join up with this GP130, and then there is signal transduction. There is also the soluble form of this GP130, and its job in fact is to inactivate this complex. So there's interleukin 6 bound to this soluble receptor on its way to carry out a signal transduction process in the cell. Instead, this guy comes in and takes it away, inactivates it. The two systems are mutually exclusive. IL-6, free IL-6 on its own, cannot take part in this signal transduction, whereas soluble receptor bound IL-6 cannot take part in this receptor system. So there's just a reminder of the leukocytes and hepatocytes having this arrangement, and all cells having this arrangement, with the soluble GP30 able to take away the complex. Notice that soluble GP130 cannot bind the free interleukin. It can only bind the receptor bound interleukin. IL-6 in patient outcome. In this study, non-survivors had higher levels of IL-6, where the survivors had lower levels of IL-6. Similar results here. Severe disease, non-severe disease. This is the IL-6 levels of the patients with severe disease, IL-6 levels of the patients with non-severe disease. This is a study of the patients with severe traumatic brain injury. So these people are looking at lots of interleukins and substances in patients with severe traumatic brain injury. Some of these patients can go on to develop acute respiratory distress syndrome, ARDS, and some do not develop ARDS. The IL-6 level compared to normal is higher in both groups, but more higher in patients who get ARDS as compared to those who do not get ARDS, but still quite high. If you look at IL-8, similar pattern, not as high as IL-6, but it is there, increase in number. ARDS have higher IL-8 compared to no ARDS, but both have higher IL-8 as compared to normal controls. In this study, I'm a bit concerned to start with because their control IL-6 and IL-8 concentration seems to be quite high. 38, 51 is a bit too high for normals, in my opinion. But when the patient has ARDS, so this is patients with acute respiratory distress syndrome, during the disease and during recovery. During the disease, there's a higher higher levels of IL-6 and IL-8. During recovery, they're coming down. When you look at the survivors and non-survivors, the survivors have a lower values of IL-6 and IL-8. Non-survivors have higher values of IL-6 and IL-8. In another study, severe and moderate coronavirus disease, IL-6 levels, this is the range, and IL-8 levels, there, these are the arrow bars, and here. IL-8. Many cells can be stimulated to produce IL-8 by IL-1, by TNF, IL-6, interferon gamma, lipopolysaccharide, and so on. The potent inhibitors of IL-8 production include dexamethasone, interleukin 4, and interleukin 10. CXCL8 is in fact another name for IL-8, earlier name for IL-8. So IL-8 can be expressed by several cell types: monocytes, endothelial cells, which are of concern to us. We will be talking about that a bit later on. Fibroblasts. Remember we were talking about fibrin deposition in that patient's alveolus with the thickened septa. Fibroblasts. Epithelial cells. So lots of cells can produce IL-8. Bit difficult to look at this data, but it's better shown on the next one. They're looking at in patients with injury and so on, looking at whether level of cytokine can predict 28-day mortality. IL-8 comes out the top. High IL-8 was leading to more mortality. MCP1, IL-6 are close by. IL-8 receptor. There are two types of IL-8 receptors. There's actually a third one, but that is not. It is not thought that this is one for signal transduction. Indeed, it's thought that this receptor is actually taking up IL-8 and degrading it, whereas these two receptors are part of the signal transduction process. Loss of effects: stress fiber formation, apoptosis, protease activation, chemotaxis, proliferation, and so on. So IL-8 binds these receptors and in various combination with other receptors and so on, what else is going in the cell, you can get several effects. There are two types of IL receptors present on many cell types: neutrophils, monocytes, macrophages, endothelial cells. Here is the membrane-bound receptors on neutrophils. PMN is a neutrophil and it's got membrane-bound IL-6 receptor. Remember we were talking about soluble IL-6 receptor? And there's one way of getting it. In comes IL-8, signal transduction, leading to events which cleaves off the IL-6 receptor from the membrane of the neutrophil. So you have increase in soluble IL-6 receptor. This is further evidence that IL-8 does it. Control neutrophils, 100% of them have IL-6 receptor on their surface, but if you give them interleukin 8, surface IL-6 receptor decreases. And what's happened is, in the supernatant, IL-8 soluble receptor increases. Implication is, from surface, IL-6 receptor has gone into the supernatant. Back to the future. Well, I'm going to go back in history, see what we can find with what's been going on. And I love time travel. So there I go, back to the future. HUVEX, I meet HUVEX, human umbilical vein endothelial cells, growing in culture. Data is here, the year is 1997. I'm going to get straight to the point, conclusion of the results. If you give the HUVEX IL-6 plus IL-6 soluble receptor, they then give out more IL-6, they give out IL-8, they give out MCP1. Note, in this experiment, soluble receptor alone does not induce IL-6, IL-8 or MCP. You have to give IL-6 also. This is in contrast to the experiments done by other workers, which I will be talking later. Not much difference, but there is some difference. Broadly, they are on the same track. 1999. Hypoxia induces IL-6 and IL-8 synthesis in HUVEC. 1996. Thrombin stimulates production of interleukin 8 in human umbilical vein endothelial cells. Thrombin, give him HUVEC thrombin, they give out IL-8. 1997. HUVEC expects some IL-6 constitutively in addition of soluble IL-6 receptor. They give out IL-6, they give out IL-8. TNF, they give out IL-8. In this experiment, remember, soluble IL-6 receptor was good enough. What they said was these cells are producing interleukin 6 by themselves. So these are HUVECs growing their own interleukin. So these cells have their own interleukin from HUVECs themselves, and soluble receptor will react with interleukin produced by these cells and then get them to secrete even more interleukin 6. TNF produces interleukin 8. So this is the summary of those results. So this is just a summary slide of what we've been seeing about HUVEC experiments. in the 20th century. But do these experiments relate to lung endothelium? We are talking about respiratory aspects of COVID-19. Lots of other aspects, but we are concentrating on respiratory, lung physiology, lung pathology, lung pathophysiology. These experiments need to be done with lung endothelium. If they haven't, they may be there, results may well be there. If they are not, they need to be done. And maybe they need to be repeated with today's technology. Their answers may be there in databases. They need to be brought to the front. (1990) A49 pulmonary epithelial cells. So it's a cell line representing pulmonary epithelium. These cells retain features of type 2 alveolar epithelial cells. Conclusion of these results is that A49 epithelial cell line, TNF or IL-beta induce IL-8 mRNA. Secreted IL-8 was also detected. So they produce mRNA plus the protein. Year 2000. Lung fibroblasts. Those fibroblasts again, the ones that produce a lot of fibrin in the lung. Although we are not talking about this aspect in this talk, this fibrin causes a lot of problem for our patients. So these lung fibroblasts. Thrombin increases IL-8 mRNA expression. And also it induces IL-8 protein secretion. That's in the year 2000. 1994, 20th century, back again. We're going backwards and forwards in this 20th century. A lot is happening there, isn't it? Baseline IL-6 production by rat type 2 pneumocytes is 5 times more than rat alveolar macrophages. So the pneumocyte type 2 spontaneously produce a lot more interleukin 6 as compared to the macrophages in the lung. If you give them IL-1, pneumocytes produce even more interleukin. Macrophages produce more, but nowhere near as pneumocytes, pneumocyte 2. Let's read that again, because that's quite a dramatic finding from archaeological look into history. Baseline production of IL-6 by type 2 pneumocytes is 5 times more than baseline production of IL-6 by macrophages. A49 cell line. Back to the A49 again. Human pneumocyte 2 cell line. In the year 1994. They're given the same amount of interferon gamma, but increasing amount of LPS. So as long as there is 20 nanogram per mil interferon gamma, and you give them more and more LPS, they give out more and more IL-6. Other results, data not shown here. IL-1 or TNF, IL-6 is produced. IL-1 plus TNF, the response is additive. This is an important experiment. Culture human alveolar macrophages with LPS, lipopolysaccharide. Collect supernatant. Then you grow those, remember those A49 cell line? Human lung epithelial 2 pneumocyte. Grow them in a petri dish and you give them collected supernatant from this culture. You get a lot of, and these A49 cells then produce a lot more IL-6. If you remove any IL-1, so these may have produced IL-1, there may be IL-1 in this tube, you take IL-1 out, then you give it to the A49 cells leave them overnight, collect the supernatant, they still produce IL-6. Not as much if you left the IL-1 in it. This one IL-1 was left in it. This one IL-1 was taken out, and they still produce IL-6. Here they produced it more. This is an important result because these macrophages may be producing something that causes A49 cells to produce a lot of interleukin. And there may be a bridge between these results and our histological findings. So the macrophages here may be secreting factors that stimulate type 2 pneumocytes to secrete IL-6. They're doing it here to a cell line, which is very similar to type 2 pneumocytes. Chances are these macrophages could well be pouring things onto this pneumocyte 2 to make it give out IL-6. Maybe the substances have diffused from other macrophages to make these pneumocyte 2 to give out IL-6. This experiment is going in line with the fact that pneumocyte 2 have been shown in histological specimen of autopsy patients to be producing IL-6. This is trying to put some pathways together. Thrombin on endothelial cells produces IL-8, which will cause release of soluble IL-6 alpha receptor. This will then bind with IL-6, and then there will be further transduction leading to MCP production. There will also be acute inflammation changing to chronic inflammation. I'm not going into that today, but that lot could be happening. So thrombin, endothelial cells, they produce IL-8 that goes on to cleave soluble IL-6 receptor. This then, if you go back to the, when we'll come to the diagram in a minute, will push events away from immune cells to non-immune cells, as you will see. So here it is. Type 2 pneumocytes could be producing IL-6, which acts on endothelium to produce IL-8. IL-8 acts on the receptor on neutrophils, gives off soluble IL-6 receptor. The soluble IL-6 receptor binds interleukin 6, and although it will signal in immune cells as well, but it will allow also signal to go through non-immune cells. So in the absence of the soluble receptor, IL-6 will only be working through leukocytes. With this receptor, it can do both non-leukocytes as well as leukocytes. Okay. So we've got to find out what the cells are in the lung in health and disease, what cytokines are being produced, what are the targets for the cytokines, what are the receptors for cytokines, and this table goes on and on and on. It looks like one or more of the interferons are stimulators of IP10 and ACE2. Well, I have to say something about ACE2. I've gone through the whole talk without even mentioning ACE2. There it is, ACE2. One thing we haven't discussed of the interleukins is, are they indicators of damage? Are they contributing to damage? Are they contributing to healing? So in other words, if there is a lot of IL-6, maybe it's showing that there's damage that has caused release of IL-6. So for example, if you talk about lactate, a lot of lactate, it does cause pathology, it's harmful, but if there's a lot of lactate, it is also an indicator there's a underlying disease elsewhere which is leading to build-up of lactate. So similarly, is there an underlying disease elsewhere which is leading to build-up of these interleukins, or these interleukins are part of an integral system that respond to disease, trying to bring it under control, and in some cases they make matters worse. Thank you for listening. I do want to thank everybody who helped me make this video. I did get a lot of help from people. This video has been made in a rush. I rushed into making this. I was making a larger video project going through with the virus, structural biology, molecular biology, cell biology, and general pathology of COVID-19, acute respiratory distress syndrome, and so on, lots of aspects of it. That project was going on and on and on, never ending, and I felt that this work which I presented today was important, that I should put it out sooner rather than having to wait for the whole project to complete. So it's a rush job. I'm a learner in the process. Most of that present today, I had to learn from scratch, histopathology especially. My knowledge of histopathology was very little. In fact, this talk I've given should be taken something similar to an undergraduate student given a talk on a project rather than expert. I'm no expert. So please, I'm limited in what I've said because this is as much as I know about the subject. Hopefully I'll put out some more information in a couple of more videos to follow. And just finally, I found this poem on the internet. I'm no poet. I can't write poems like this. I can find them on the internet, but I can't write them like this. And I was never sure whether you were the lighthouse or the storm.